It's a very special privilege for me as a head of this institution which is hosting this annual meeting, and also to be a speaker in a symposium that is specially planned in this annual meeting. And we thank Rajiv for accepting our request and organizing this symposium and inviting some very special speakers, Professor Gupta ji, Swapan Datta ji, and then Professor Deepak Pental and Rajiv himself. I'm sure it is going to be a very interesting session but then it is also important for us, for some of us, to make it easier for all of you, some of you not being in biological science, to make it little easier for you to understand that this host pathogen interactions is, as Madam rightly mentioned, is very important topic, and we should understand. We all know that there is a immunity in us, in human beings, and also in mammalian systems, and you know that you can immunize you can take a vaccine and then you get immunized against infectious diseases. And many a times the plant scientists have realized this. So many things after the people in medical microbiology have realized, and it so happened in this case also it is in a similar way. I'm going to give you a little of introduction to that, and after that I will narrate to you what exactly we have been doing for the past maybe a decade or so, some of this which I will summarize. When our lab primarily is interested in using biological methods for control of diseases of plants, particularly fungal diseases of plants. We are not interested to use chemicals directly. The synthetic chemicals are not to be used is our concept, and that's where we are working on the approaches that will allow us to use biological molecules derived from biological systems or the organisms themselves for the control of fungal diseases of plants. So I will take you to the topic. You look at this beautiful picture. You will be, all of us enjoy this. When we look at this kind of a greenery, we all enjoy it. And this is all because those plants are healthy and they are happily living there. And all of you should also know that there are thousands of different pathogens around which have the potential capability to cause the disease of different plants. In spite of having several thousands of pathogens around, the plants remain healthy. This is a natural system. The plants remain healthy. It is not possible for them to remain healthy if they were not to have an immune system. An immune system which is not parallel, which the terminology wise it is parallel, but the components wise it is not similar. There is a different kind of an immunity system that operates in the plants, which we will try to examine, and a little of it I'll see. If the plants were not to be having an immunity system of their own, when thousands of different pathogens are around, what would have been, the situation would have been like this. Because one pathogen can kill one plant and it would get eliminated over a period of time. Likewise, if different pathogens would have killed different plants and all the plants would have got eliminated, we would have seen only a barren land like this. And probably if that was the situation, none of us would have been here to discuss anything about science, because there is no possibility to live without these plants and the plants being healthy. So that's the importance of the topic. Let us see. I just want to show you this, since this is a common, a general audience, I wanted to show you that by looking at these things, I want you to have an impression of your own. When you look at any of these, next time when you see, easily you recognize. They have unique physical structure, what we can call as a pattern. It has a unique specific pattern. You will not miss it. For example, this is cobra. When will anybody miss that? If you see that second time, oh, this is cobra, you will know that. It is as simple as that. And another important thing I wanted to show you here is a banana fruit. It's not for any nutritional purpose. It is just to show you a fruit. If you see this in the daytime, and even if somebody gives you in the night, you touch it from one end to the other end, you will be able to say it is banana. Even if it is completely dark, you will be able to say, because that pattern of that fruit is imprinted in our mind. And do the plants have this kind of a mechanism to remember something what they have seen before? And how do they recognize that? That's the question. And this is what I was telling you, different kinds of chemical substances that are present on the surface of the pathogens, the human beings and animal systems are known to recognize them. This has been evidenced adequately in the literature for several years, and that's where we understand about immune system. We have developed vaccines, and we are using vaccines successfully to prevent several of the infectious diseases and serious diseases as well. Here is the plant. For example, there is a fungus which is on the surface of the leaf. There are some insects on its surface. There is a bacterium which is infecting the leaf. And we are able to see an insect here, but we are not able to see the clear structure of the fungus. We will not be able to see the clear structure of the bacterium here. Nothing can be seen here. And when we cannot see, how the plant is able to see? The point is, what is actually the surface structure of these insects, fungus, and the bacterium? What is common in them? That is what is seen by the plant. The common in them is something called a polysaccharide, which is called chitin. Chitin is a molecule which is present on the surface of the insect exoskeletons and in the fungal cell wall, and also a similar polysaccharide is present in the bacterium, which is not exactly chitin. It is a peptidoglycan, we call it. So this is how the molecules that are present on the surface of the plant can recognize these molecules. These are the proteins that are present on the surface of the plant cell membrane, which have an intracellular domain. There's something inside the cell, something outside the cell. The outside extracellular domains can interact with the molecules that are coming from the microorganisms like fungi, insects, and bacteria. Here is a peptidoglycan, here is a chitin, fungus, and here is something called mycorrhizal factor. And here there is a subtle difference. There are some fungi which are beneficial to the plant, there are some bacteria which are beneficial to the plants, and there are some fungi which are serious pathogens of the plants, and some bacteria which are serious pathogens of the plants. How then, when both a fungus which has a chitin as a cell wall, is recognized as a beneficial organism, as an organism which can cause disease on the plant? How the plants recognize that also we'll try to understand. So these are the typical molecules which are evolved in the plants, where they can interact with the carbohydrate ligands that are coming from the microorganisms, and then can activate a symbiotic pathway like what we see here, where many of you know the leguminous plants have some kind of a association with rhizobia and form nodules and fix atmospheric nitrogen. And here is another symbiotic nitrogen association, what we call as mycorrhizal association. The other two being fungal. This is against fungus a resistance, and again against the bacterium a resistance. These are the different types of molecules that I am talking about which are present on the surface, but the most important one being the lysosome kind of receptor, we call it. It has a lysosome modules, extracellular modules, to which the carbohydrate ligands from the pathogen can be bound, and then accordingly the signal can be transduced inside the plant cell for the plant to respond. That is the lysosome module. Yeah, I was telling you that how the pathogens and symbionts are recognized. Probably there is a hypothesis, now there is a hypothesis which is coming forward for the last maybe 5-6 years or a little over 6 years. The hypothesis is, if the carbohydrate ligand has a lipid component, then we call it as a lipid chitin oligosaccharide. If the carbohydrate ligand has a lipid component, that's called lipid chitin oligosaccharide. And the more of the lipid component on the chitin oligosaccharide, the pathway that is activated inside the plant will be symbiosis. That is what is known in rhizobium, and also what is known in the mycorrhizal factor, that there is a lipid component attached to the chitin oligosaccharide, which you don't find in a chitin oligosaccharide which is originating from a fungus which only causes a disease, or a bacterium which only causes a disease. There is no lipid component attached to that. So this probably is the distinction that people are working with, this hypothesis that the more of a lipid component, it will be a symbiotic pathway. No lipid component, it would be a pathogenic, a kind of a disease or resistance pathway that will be operated. I'll skip this. Yeah, this I want to introduce to you. If chitin, for several of you are being chemists sitting in this room, chitin is a crystalline molecule, polymer, which is not easily soluble. When it is present on the surface of the plant, such an insoluble molecule can never be recognized by a biological system. And during the interaction, the plants are known to secrete molecules like chitinases and endokitinases, exokitinases, gluconases, different types of enzymes, which solubilize this wall and thereby release what we can call as oligomers. Oligomers of the chain length somewhere between 8 to 10 or even larger than that. But the most soluble ones and the biologically active ones that have been characterized in the literature are the octamers, which is a repeated unit of N-acetyl glucosamine, 8 units of N-acetyl glucosamine, we call it as octamer, or this is the configuration which will interact with this receptor molecule on the surface of the plant and induces what we call as dimerization of the receptor, and that is the signal for the receptor to transduce the message right into the inside the plant and activate either the immune pathway or the symbiotic pathway. Well, this is the source of chitin I wanted to show you. The shrimp waste. Several of us are love to enjoy this shrimp, and plenty of waste is generated when we process this shrimp. And this waste is a rich source of chitin, which can be used if it can be produced into some kind of a soluble molecule whose unit length is 6 to 8 units. That it should be possible for us to make use of it for protection of the plants, to protect the plants against the diseases. That's the important issue. And this is an insoluble chitin that you can easily prepare from that shrimp chitin, which is shrimp waste, which is very, very least expensive material, I would say, not more than 200 rupees a kg that it costs. It's not that expensive, and it is, since it is biologically synthesized, it is biologically degradable as well. And a few years ago, we were working on bacterial chitinases, and then we realized that this bacterial chitinases, which are supposed to do only a hydrolysis, were also doing something beyond hydrolysis. What we call, if a hexamer, like this DP6, when I say it is a hexamer of chitin, and this hexamer, when it is broken down, what we should see is a pentamer, tetramer, trimer, dimer, and like that when it is hydrolyzed. But in addition to that, we also saw some other molecules longer than the substrate. Similarly, when we take a pentamer, we see longer than pentamer, and you can see more clearly here when we take a trimer, you get a dimer and a monomer, and you also get a tetramer, pentamer, hexamer, that's longer molecules. So this enzyme was doing something more than hydrolysis, what we call it as transglycosylation. To put it in a simple term, this is transglycosylation, where the enzyme is able to take the smaller oligomers into it and then add a glycosidic bond and then synthesize a longer chain oligomer. And this is what is interesting for us, because most of the time the enzymes or acid hydrolysis of chitin can generate a very small molecules of chitin oligomers. Dimers, trimers can be easily produced. Can we convert the dimer trimer into a longer molecule which can activate defense in the plant? That's where was our interest. And these are all characterization, and we try to improve the enzyme, making use of some mutagenesis. We have collaborated with a chemist, with a colleague in chemistry, that is Professor Lalita Guru Prasad, and identified some residues in the amino acid residues in that enzyme, and we went on to improve the activity, improve the yield of these transglycosylation products. That means longer chain oligomers we are able to produce more by carrying out mutagenesis. That's what we see. And another important feature that we see is here we find that these modified enzymes or the mutants are able to produce these molecules, transglycosylated molecules, that is longer chain molecule, for a long duration of time compared to the wild type mutant. And this is a summary of all that, and that was our first solid paper that we tried to show what is all this application of this enzyme and how this enzyme is working. And beyond that, we went on to do several modifications to that enzyme, and we tried to understand, again through collaboration, we have done some molecular dynamic simulation studies, and we tried to identify what is the entry site of the substrate, what are the exit site of the molecule product in that enzyme, and with the hypothesis that if we introduce a bulky amino acid residue at the exit site, the products will be retained for some duration inside the reaction tunnel. And by introducing those residues, we are able to, once we map the exit point and the entry point, When we introduced a bulky residue in the entry point, we could see that there is a drastic reduction in the transglycosylation, and in the exit point, when we have introduced a bulky residue, there is a significant increase in the transglycosylation, thereby increasing the yield of these transglycosylation products. This is all the analysis of all that. This is a summary of it. When we introduce in the entry point, you can see that there is a significant loss in the transglycosylation products, and here you see a significant increase from what you see in the wild type. There is a significant increase in the transglycosylation products. And we tried alternative approaches further to improve the enzyme activity and to increase the yield of these products. And these are all through typical biotechnological tools, combining the different domains which can help the enzyme to do the reaction much better. We have done all that. We have also identified, we have collaborated with Professor T.P. Singh at Royal University of Medical Science, and then worked out the structure of this protein to understand the structural features of this protein. And then we realized that there is a very interesting loop region which is there in the upper and lower half of this protein. And this loop region plays a very crucial role in the transglycosylation that also we have established by doing again some kind of a mutagenesis. We have clearly established that this loop region does play an important role in transglycolysis. So the last part of it, what I would like to convey to you is that these molecules, this is the enzyme, these are the wild type enzymes and mutant enzymes. With the substrate, when we incubate, we generate a mixture of, I have not made it complicated because the chemistry people are there. You may be tempted to ask me, there are a lot of complications in the chemistry of the chitin, but I have not complicated it for the common public. There are some acetylated residues and deacetylated residues, because when a portion of these glucosamine residues are deacetylated, we call it as chitosan. We don't call it anymore chitin. So the chitosan has acetylated and deacetylated residues in it. When we incubate the chitosans with these enzymes, we generate a mixture of these molecules and we test them on the suspension cultures of the plant cells. We see that there is an activation of defense response in the plant. That's the most of it, how we have generated these molecules and what is the mixture of molecules that we have there. And we try to test the molecules which are only longer chain oligosaccharides and not the shorter ones. And when we tested these oligomers, which are long chain oligomers, we find that there is a dose dependent elicitation of oxidative burst in the rice cell suspension cultures. And this, of course, again, there is a little more complication. If I don't know whether I'm close to the time. The chitosans, you have, depending on the degree of deacetylation, we define it as DA, degree of acetylation, 61, lower degree of acetylation, higher degree of acetylation, medium range degree of acetylation. And beyond this, now, in fact, we have not yet published, it is already submitted to JACS. There is something beyond the degree of acetylation that is influencing the activity of these oligomers, what is called as pattern of acetylation. That we have collaborated with a German group, and that work is coming up hopefully sometime in this year in JACS, it should come. So these are the molecules with different degrees of polymerization from 6 to 11. We have tested all of them, and these fractions were generated by the wild type enzyme and the mutant enzyme at 61 degree of acetylation, 61% degree of acetylation. Now we have optimized the process. We are able to produce little higher quantities of these oligosaccharides, and we are now testing these oligosaccharides on plants, directly onto the plants as spray. There are two different approaches we follow. One of it will be direct spray of these oligomers, which will be a little more expensive, and we are trying to use a nano-based method to deliver these oligosaccharides into the plant system. The two different approaches that we are following, and we are able to see that there is a foliar treatment induces elicitation response early, and it is sustained for a long duration of time. So my dear friends, I hope I have brought an important concept into your mind that the plants do have immunity of their own, and they recognize the plants recognize the pathogens, particularly the microorganisms like fungi and bacteria, based on the surface carbohydrate chemistry of the fungus and the bacterium. And it is not the polymeric molecule that is recognized, it is the soluble oligomeric components that are recognized. and there are specialized receptors that are lined up on the surface of the plant cell membrane, which can interact with these oligo or ligands that are coming from the pathogens differently, and then can activate different responses from inside the plant. There is much more complication, like we call it as pathogen associated, or PTI we call it as, and there is another immunity we call it as ETI. There are two different types of immunity which I have not complicated for you. I thought it will become too much, and this is all I wanted to share with all of you. And beyond that, we have been substantiating all that work and then trying to understand more about this enzyme and as to how this enzyme can be made use of further to enhance the yield of these oligosaccharides and also produce better oligosaccharides for application onto the plants for immunity. So these are my collaborators and the students who have contributed for this. Lalita Guruprasad is in School of Chemistry in University of Hyderabad. Professor T.P. Singh, many of you would know, at All India Institute of Medical Science. Bruno Morsbacher at University of Munster. These are all my students, Purushottam, Suma, Subbanarayan, Madhu Prakash, and Manjit, and then Paparao and Rambabu. And this is the recent group that we are all a group at the School of Life Sciences. And I thank all of them. And lastly, I also thank the Government of India for the support that is given to us in the form of different type of research grants. Thank you very much.